Assalamualaikum warahmatullahi wabarakatuh. So we will start in another 2 minutes. Don't forget to fill your attendance, and the link is in the comment section. Don't forget to fill your attendance, and the link is in Hi, Assalamualaikum warahmatullahi wabarakatuh. So welcome to this live stream session. So today we are going to discuss on Bio091 Foundation Biology 1, and we will discuss. We will have a very quick revision on chapter 3, 4, 5, 6, 7, and 8, and then we will discuss the past year questions that you have answered in the paddock given. Right, so let's, without further ado, let's start our quick revision on chapter 3 until chapter 8. If you have any question, you can put your question in the comment section. All right, so I probably will read your question and will answer it. And don't forget to fill your attendance, and you can have the Google form in the comment section also, right? Right, so let's start with revision on chapter 3, plasma membrane transport. So basically in chapter 3, in plasma membrane transport, I prefer to divide it into two sections, which is the first one is the structure and function of plasma membrane, and the second part is the transport across plasma membrane. So this basically the structure of plasma membrane, that it includes several elements. I think you have heard of them before. So we have protein, that is peripheral protein and integral protein. And then you also have carbohydrate that is attached to the lipid, and it is called as glycolipid. We also have carbohydrate, which is oligosaccharide that attached to protein, and we call it as glycoprotein, right? And outside of the plasma membrane, we have extracellular matrix, okay? And basically, the plasma membrane is called as fluid mosaic model, and it is due to the membrane fluidity and the mosaic structure by the protein itself, okay? So your membrane is fluid because it can move. So there are two types of movement, flip-flop and lateral movement. Right, so in the second part of plasma membrane, you learn about transport. Right, so in transport, okay, in transport, you learn about passive and active transport, whereby in passive transport, you learn that it is comprises of simple diffusion, facilitated diffusion, and simple diffusion is divided into two, that is in simple diffusion, you learn about osmosis, that it is transport of water across plasma membrane without using any transport protein, right? And in facilitated diffusion, it is the transport across plasma membrane without using any ATP. The transport is following concentration gradient. However, it need transport protein. Okay, so there are two types of transport protein used: carrier protein or channel protein. So in channel protein, I used to divide it into two types of channel protein. One is non-gated, the other one is gated channel protein. And you need to bear in mind that carrier protein change shape. However, channel protein do not change shape, right? So carrier protein will change shape, and it is usually due to the binding of substance or due to the binding of phosphate group, right? However, channel protein do not change shape. Okay, so channel protein, even though it do not change shape, it can have gate. It's just like your house, okay? Your house is not changing in shape, but you can open your gate or close your gate, okay? So in this case, channel protein ada gate, ada yang tak ada gate, right? So the one that is non-gated channel protein is always open, right? However, the gated channel protein, in order for you to open the gate, you need to have a correct key. So the correct key here is the stimulus. So we can have voltage as the stimulus, ligand or mechanical as the stimulus, right? So next one, I think you learn about active transport, that it need ATP, and it transport against concentration gradient. And you learn in active transport about electrogenic pump, co-transport, as well as bulk transport. So in electrogenic pump, you learn about sodium potassium pump, that you will transport 2 sodium, and on the other hand, kita transport 3 sodium and 2 potassium. Okay, and this electrogenic pump is basically to this electrogenic pump is basically to generate voltage gradient across membrane, So meaning that we wanted to maintain the gradient. Okay, let's say inside of your cell is always negative, outside of your cell is always positive. So we always wanted the different concentration. We wanted to maintain that different concentration. We don't want equilibrium to achieve. That's why kita guna active transport, right? So we also have proton pump, okay, which is another example of electrogenic pump. All right, I think you are familiar with electrogenic pump, which your electron transport chain is one example of electrogenic pump, that it pump electron from one protein to another protein, and then it transport H plus together. Okay, regardless dia transport keluar atau ke dalam, but it transport H plus. The energy released by the electron, okay, it's not in the form of ATP, tapi the energy released by the electron is used to transport H plus across the membrane, right? And kenapa kita transport H plus across the membrane? Kita memang nak maintain the different concentration. Okay, that is in electron transport chain. Regardless dalam photosynthesis atau cellular respiration, right? Next one is you learn about co-transport, that kita transport one substance with another substance, whereby the other substance is basically transported secara passive. Alright, so in this case, glucose is transported against its concentration gradient. However, Na plus is transported following its concentration gradient. Okay, so this is co-transport. Similarly, sucrose is also transported against concentration gradient. However, H plus is transported following concentration gradient. Next one is, lastly, you learn about transport, that we transport a certain substance into the cell or out of the cells in bulk. secara banyak, bukan satu molekul tu kita transport banyak-banyak. Right, so endocytosis is when you transport into the cell, because endo means dalam, so you transport the substance into the cells. And exocytosis is when you transport substance out of the cell. Right, and if you see here, kenapa saya warnakan warna oranye ni, okay, kerana in this chapter you learn about 3 types of disease that related to these 3. Okay, you learn about cystinuria, cystinuria that related to carrier protein, cysteine carrier protein. You learn about cystic fibrosis that related to Cl- channel protein. Okay, cystic fibrosis. And lastly, you learn about hypercholesterolemia that is related to receptor mediated endocytosis. So please make sure that you know this. Okay, kalau you rasa masih tak faham pasal benda ni, I think you need to read more, or you can ask question in the comment section. Right, next one, let's move on to chapter 4, which is enzyme. So in enzyme, you learn about its characteristic. I think you are familiar with this. Enzyme is protein, usually globular protein, can be reused, blah blah blah. So that is characteristic of enzyme. And then you learn about mechanism of enzyme. We have induced feed mechanism and lock and key mechanism. Basically, both are nearly the same. However, in induced feed mechanism, the active site is not exactly kita kata is flexible atau is not exactly similar to the substrate. Okay, in induced feed, the active site is flexible, that it can slightly change shape to fit the substrate. However, lock and key, in lock and key mechanism, the active site is exactly complementary to the active site. And the rest are nearly similar lah, cuma dekat shape of active site saja that they are different. Right, next one, you learn about Michaelis-Menten and Line-Weaver-Burg. So in this part, you need to know how to read the graph, okay? How to find the Km value, 1/Vm and so forth, right? Next, you learn about cofactor, that we have 3 types of cofactor: coenzyme, prosthetic group, and metal ion. Okay, I think in 2018, I guess, we have this type of question regarding cofactor, right? And regarding inhibition, we have a reversible inhibitor and non-reversible inhibitor. That untuk non-reversible, maksudnya the enzyme is damaged, okay, because the inhibitor bind permanently to the enzyme. It's either destroy the active site atau it bind to the active site itself, so that the substrate cannot bind to the active site anymore. Right, in reversible inhibition, we have 2 types of reversible inhibition. They are competitive inhibition and non-competitive inhibition. Which competitive, the inhibitor, in competitive inhibition, the inhibitor is similar to the original substrate, that it compete for the active site. However, non-competitive inhibition, the inhibitor is not competing for the active site, but it bind to the allosteric site, and then it changed the conformation of the active site. So basically, competitive inhibition can be overcome by adding more substrate. However, non-competitive inhibition cannot be overcome. Right? And you learn about regulation of enzyme activity. So we have allosteric activator, example is cooperativity. And we also have allosteric inhibitor, example is feedback inhibition. We learn about classification. I'll show you in the next slide. And then we also have enzyme immobilization. So I think this immobilized enzyme is also frequently asked in the exam. Usually it will ask in the exam because it's a type of application. So usually this type of question you akan jumpa dalam soalan, saya rasa soalan chapter 4 tu dekat hujung-hujung lah, dekat soalan D ataupun soalan C macam tu. Right? So ni adalah graf yang you mungkin kena biasakan diri. Ni adalah graf concentration of substrate, concentration of enzyme juga, graf dia macam ni juga. Then we have temperature, and ni adalah graf untuk pH. And this is the cooperativity example of allostery in activation, and this is the example of allostery inhibition. So untuk allostery inhibition, you should know that the end product will bind to the allosteric site and it will change the shape of active site. So when we have higher product, kita akan activate the feedback inhibition mechanism. Kalau produk tu rendah, tak boleh nak buat feedback inhibition because not enough product to bind to the allosteric site, right? So let's look at immobilized enzyme. So untuk immobilized enzyme, kita ada beberapa cara. I think the first one is adsorption, okay, and then we have entrapment. Untuk binding, kita ada dua type of binding, covalent binding and cross-linking, and lastly we have encapsulation. So I think what you need to know is the description of each technique. Okay, in adsorption, it actually bind in as weak bond. These are the example of weak bond, right? And the bond is formed between the inert support and the enzyme. Right, so inert support maksudnya support tu tak memberi apa-apa kesan pun kepada chemical reaction of that enzyme, right? So untuk entrapment, we trap the enzyme in 3-dimensional lattice, right? So it is not chemically bound. So example is alginate or silica gel. So I think ni adalah contoh immobilized enzyme in the bits of alginate gel. I think in the year 2018 kot ada soalan tentang this type of immobilized enzyme immobilization. Dan soalan tu tanya kenapa kita guna alginate gel, right? Biasanya sebab dia adalah inert lah, sebab dia tak memberi kesan kepada chemical reaction. So the enzyme is also not chemically bound because you trap them in the alginate bits, right? And next one is binding. Saya kata ada dua, covalent bonding and cross-linking. And finally is encapsulation, that it is wrapped in semi-permeable membrane. Alright, sorry. So let's move on. Okay, so let's move on to chapter 5, cellular respiration. I think if you have any question, boleh tulis di comment section. Oh, ada soalan kot. Oh, tak salah. Okay, if you have any question, please write down in the comment section. Waalaikumsalam. Alright, so let's move on to chapter 5, cellular respiration. I think for my own class, I guess they are familiar with this table. This is the table that I usually use in my class. That it consists of the process for cellular respiration, the location, product that it form, and the number of ATP formed. Right, and I divide the process into 3 process, which I exclude the oxidative level phosphorylation. Okay, kenapa saya exclude? Sebab oxidative tu saya dah kirakan dia punya ATP tu di sinilah. Alright, tapi sebenarnya dia ada, cuma saya dah kirakan di bahagian calculation tu. Right, so for process, we have glycolysis, the first process of cellular respiration, followed by pyruvate oxidation and Krebs cycle. So glycolysis takes place in cytoplasm, pyruvate oxidation takes place in mitochondria, and Krebs cycle takes place in mitochondria matrix. So oxidative level phosphorylation will take place in cristae atau mitochondrial membrane juga. Okay. So in glycolysis, we basically produce 2H2O, 2 NADH, and we produce 4 ATP, 4 ATP net 2 by substrate level phosphorylation. Right, so I will always encourage you, if question asks how many ATP is produced in glycolysis by substrate level phosphorylation, I always encourage you to answer 4 ATP. Always answer it with 4 ATP and then followed by net 2. Right, sebab berapa banyak ATP dihasilkan? Memang 4. ATP yang dihasilkan memang 4, tapi kita kata ada 2 je sebab kita tolak dengan penggunaan dekat energy investment phase. Right? So I will always encourage you to answer 4 ATP net 2. Tapi kalau dalam objective question, kalau dia bagi jawapan, saya harap, saya tak rasalah jawapan tu akan mengelirukan you lah. Sebab jawapan 4 dan 2 tu kedua-duanya adalah betul. Okay? Depending kepada konteks jawapan yang diberi lah. Okay, right. And then let's look at saya tak tunjuk pengiraan dulu ya. Saya tak tunjuk pengiraan dulu. So let's look at the mitochondria. In pyruvate oxidation. In pyruvate oxidation, it will produce CO2 and NADH. Okay, and tak ada ATP diproduce di sini. Right, hanya ada NADH saja. Right, and next in Krebs cycle, so it will form 6 NADH, 2 FADH2, and it will form 2 ATP by substrate level phosphorylation. So in this case, it is solid 2 ATP lah. Right, and these are the calculation that taking into account one glucose molecule. So if soalan tanya kalau dah oxidize, fully oxidized of one glucose molecule, so this will be the answer, right? Kalau dia kata 2, 3, 4 glucose molecules, then you have to multiply that. Okay, and so this is what happened in cellular respiration. And next we have oxidative level phosphorylation, where in oxidative level phosphorylation is divided into electron transport chain and chemiosmosis. Okay, and I think you know that this is electron transport chain and this is chemiosmosis that involve ATP synthase. Right, and I hope you know that this is matrix mitochondria and this is intermembrane space. Right? Okay, so what happened in oxidative level phosphorylation is we have our electron carrier, NADH and FADH2. Both of them will donate the electron to the electron transport chain, okay, to the protein complex, and the movement of electrons, the electron will move atau kita kata will Hi, sorry, can you hear me? All right. Okay, before I continue, let's read some of the questions. Medong akan save ke live ni? Save code, I think dia automatically save code. Alright, sekejap citric acid cycle Saya tulis dua citric acid cycle. Sekejap. Let me update this. Oh yes, betul. In Krebs cycle, we have another product which is 4 CO2, correct. So let me correct this one first. For CO2. Right. Okay, let's see this question. If question asks how enzyme lower activation energy, what info do we need to include in the explanation? Do we need to describe the F and the transition state, or do we need to include other info? How enzyme lower activation energy? Okay, good question. Let me share with you the enzyme slide. Okay. Oh, sorry. Okay, so let me share with you this slide to answer your question. Right. So this is what you need to write. Okay, so you can write down, it have the correct orientation of substrate. It have favorable microenvironment, bond in reactant stretch to achieve transition state, and it form temporary covalent bond with the substrate. I think for my class, I have shared with you the videos on how enzyme activity from YouTube, which I shared to you in the app puzzle. Okay, so this is that video is basically to explain this mechanism. How does the enzyme lower Okay, so I think this is the answer. Saya letak dalam komen ya. F2 free energy tau. How ATP produced in oxidative phosphorylation? Okay. Soalan Dik Nurfana saya dah tak saya baru tak sorry. Okay, let's see here. Explain how ATP produced in oxidative phosphorylation. Okay. Okay, baik, saya akan explain. Sebenarnya saya tengah explain tadi, tiba-tiba hilang pula Saya explain semula tentang oxidative phosphorylation. Hai guys, Assalamualaikum. Sorry, kita hilang lagi connection ni sekali lagi. Hopefully kali ni okey lah. Can you all comment? Ada lagging lagi ke? Can you hear me clearly? I was actually about to answer this question. How ATP produced in oxidative phosphorylation? Okay, so I think I'll share the PowerPoint first. Okay. All right, so let's move on here. Sorry for the interruption. Hopefully the internet is okay now. Okay, so oxidative level phosphorylation is basically one of the way to produce ATP. So first of all, you need to understand what is phosphorylation. This is the first thing that you need to understand, that phosphorylation is the adding of phosphate group to any substance. To any substance. As long as you add phosphate group to any substance, you call it as phosphorylation. Okay, in this case, we are actually adding phosphate group to ADP and we will form ATP, right? So this is phosphorylation in this context dalam cellular respiration. Okay, sebab kita nak buat ATP, so phosphorylation adalah adding phosphate group kepada ADP to form ATP, right? So next question is, siapa yang add dia? Siapa yang catalyze the reaction? The answer is ATP synthase. ATP synthase is an enzyme and it will catalyze the addition of phosphate group to ADP, so it will form ATP. Betul kan saya cakap? Phosphate ATP synthase catalyze, okay, the formation of ATP. Macam mana? Dengan adding phosphate group to ADP, right? So kat mana dia berlaku? Dekat kristal. Okay, dia berlaku dekat kristal lah sebab ATP synthase berada pada kristal. Right? So in oxidative phosphorylation, we divide this process untuk kita bentuk ATP ni, kita divide proses oxidative phosphorylation ini kepada dua. Electron transport chain dan chemiosmosis. So kenapa kita panggil oxidative? Alright, so basically phosphorylation ni berlaku pada peringkat chemiosmosis lah sebab kita add phosphate group kepada ADP. Now oxidative ni berlaku dekat mana? Oxidative ni merujuk kepada the redox reaction, oxidation and reduction. Okay, so this redox reaction occur at the protein complex. So kita ada banyak protein complex and this protein complex akan receive electron and it will donate the electron alternately. Right, satu receive and then dia donate and then yang lain receive, dia donate balik. So it is the redox reaction continuously occur. So kita panggil proses ini sebagai oxidative phosphorylation. Eventually, finally kita akan produce ATP. So apakah kepentingan oxidative part ni? Apakah kepentingan electron transport chain kepada chemiosmosis? What is the significance of electron transport chain to the chemiosmosis? Significant dia ialah dia akan maintain electrochemical gradient. So please remember that atas ni adalah intermembrane space. I think dalam gambar aja dia boleh jadi atas, dia boleh jadi bawah lah, mana-mana satu. Macam mana nak identify which part is intermembrane space adalah dengan melihat arah H+ pergi. Sorry, arah H+ lalu. Right, so H+ is always moving, is always pumped from matrix mitochondria to intermembrane space. Tak nampak tulisan. Dia akan always lalu dari matrix mitochondria kepada intermembrane space. Sorry, tulis balik ya. So this is matrix. So this is intermembrane space. Okay, so intermembrane space atau matrix ni boleh jadi atas atau bawah, mana-mana satu saja bergantung kepada gambar rajah yang diberi. So to identify which part is intermembrane space, tengok kepada movement of H+, right? So this electron transport chain is important to maintain the electrochemical gradient, which we always have high H+. We always have high H+, concentration in the intermembrane space, and we have low H+ concentration dalam mitochondria matrix. Alright, so basically untuk passive transport selalunya kita akan the substance akan move from high concentration to low concentration until equilibrium is achieved. Okay, tapi untuk active transport, we don't want equilibrium to achieve. We want the different in concentration. Kita nak maintain different in concentration. So we wanted to maintain high H+ in the intermembrane space. Kenapa? Sebab high H+ in intermembrane space akan diffuse into matrix. It will diffuse into mitochondria matrix. Alright, so as H+ diffuse into mitochondria matrix, it will move in via ATP synthase. So remember, dia akan masuk ke dalam masuk ke dalam ATP synthase, the rotor akan spin and the catalytic knob akan activated. So as the catalytic knob activated, the ATP synthase is said to be activated also. So it will catalyze, don't forget the word catalyze the addition of phosphate group to ADP, forming ATP. Right? So basically electron transport chain is needed to form ATP. Kenapa? Sebab the electron that is donated by two electron carrier, which are NADH and FADH2, will move from one complex to another complex, right? And the energy, as it moves, the energy is released. So the energy that is released is used to transport H+, kita panggil transport, bukan diffuse. It will use to transport H+ ni transport H+, atau kita kata pump H+ from matrix to intermembrane space. Right? So this will create proton motive force. Right? And finally, electron will be received by oxygen atom, and it will add with H+, forming water. And it will combine with H+, forming water. Okay, so basically oxygen atom is final electron acceptor. Right, so far boleh faham? I think it is similarly to photosynthesis, cuma banyak soalan Okay, kejap saya explain. Cuma saya kata tadi oxidative phosphorylation is similar dalam photosynthesis juga, cumanya untuk photosynthesis kita panggil sebagai photosphorylation, right? Sebab phosphorylation masih berlaku, which we also have chemiosmosis. Kita juga ada electron transport chain dalam photosynthesis. However, the source of electron tu datang daripada water, H2O, which H2O tu only split bila photosystem tu dah empty. Bila photosystem tu empty, H2O akan split untuk donate dia punya elektron. Right? Yeah. Okay, so in order for the photosystem tu untuk donate elektron, kita kena ada light that strike. So kita panggil dia sebagai photophosphorylation, right? But however, photophosphorylation is nearly similar to oxidative phosphorylation. Cuma beza dia adalah kepada the source of electron. Siapa yang bagi elektron? Which in plant, the one that donate electron is H2O. Dalam cellular respiration, yang donate elektron adalah glucose. Alright, and then untuk photophosphorylation, dia occur dalam chloroplast. Tapi untuk oxidative phosphorylation, dia occur dalam cellular mitochondria. So photophosphorylation pula, H plus is transported daripada stroma into stroma into intermembrane space ke? Yang ni kita transport dari matrix to intermembrane space. Sorry, untuk photosynthesis dia transport daripada stroma into thylakoid space. Minta maaf salah cakap. Okay, so I think let's look at the questions in the chat. Apa soalan ni? Okay, soalan yang ni saya dah explain. Different between phosphorylation and photophosphorylation. Different dia adalah pada source of electron, location and organelle dia. Next one. Okay. Next one. Apakah soalannya? ATP synthesis dalam chemiosmosis tu substrate level phosphorylation ke? Tak. Okay. Saya akan jawab soalan ni menggunakan gambar rajah. Sekejap lagi Saya jawab soalan dulu. NADH and FADH2 release the why NADH and FADH2 release electron at different point. Okay. So I'll answer this two question. Saya buka PowerPoint slide dulu untuk plus chapter apa cellular respiration kan? Okay. Macam mana kita nak bincang pasti question ni? Okay, so let me share with you this one. I'll answer that two question. Okay. Basically, we have three types of phosphorylation. Inilah. Okay, inilah. Okay. Boleh nampak kan? Sorry. I'll hide myself first. Okay. Hide this one too. Alright, so let's look at here. Okay, so basically we have two types of tiga sebenarnya, three type of phosphorylation, which is oxidative, saya padam balik. Three types of phosphorylation. One is substrate level. Second one is oxidative phosphorylation. The third one is photophosphorylation. Alright, so substrate level phosphorylation adalah addition of phosphate group to another substrate. Addition of phosphate group from one substrate to another substrate. So that is substrate level. Kalau oxidative level phosphorylation ni yang you nampak tadi, you ada redox reaction that will result in electrochemical gradient of H plus, and then H plus diffuse into ATP synthase. So inorganic phosphate will be added to ADP. So it will form ATP. So that is oxidative phosphorylation, which you menggunakan redox reaction on the first place so that you dapat energy that is released from electron, and then you create concentration gradient, and then H plus akan diffuse it. So that is oxidative phosphorylation. Photophosphorylation is the one that occur in photosynthesis. So in substrate level phosphorylation, you can see here, this is phosphate group. Salah. Phosphate group is from one substrate dalam gambar ni yang atas sorry. Right, so tengok dekat sini, this is phosphate group from 1,3-biphosphoglycerate. It will be added to ADP forming ATP. So you dapati hasilnya akan ada 3-phosphoglycerate, which this phosphate group is missing. Mana dia pergi? It has been added to ADP to form ATP. So this is substrate level phosphorylation, which the phosphate group daripada substrate masuk ke dalam substrate yang lain. Dimasukkan ataupun dipindahkan kepada ADP so that it will form ATP. What is the name of enzyme that catalyze this reaction? Contohnya adalah phosphoglycerokinase. Okay, ada banyaklah contoh enzim. Biasanya enzim yang kinase ni dia memang akan terlibat dalam 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 transferring functional group. Okay, so in this case pun sama juga, we have phosphate group. This phosphate group is added to ADP to form ATP. So pyruvate tak ada phosphate group. So this is substrate level. Daripada satu substrate pergi kepada substrate yang lain. Oxidative level tadi you dapati dia kena lalu jalan yang jauh. Dia kena ada redox reaction first. Okay. Hopefully you are clear now. Next adalah soalan kenapa NADH and FADH2 release electron at different protein? Jawapan dia sangatlah mudah. Cuma I think I will ask you to look back to watch the video dalam YouTube channel yang menjelaskan tentang diagram ini. Right, I think it is very clear dalam YouTube channel tu dia dah cerita dah macam kenapa NADH dan FADH2 donated pada kompleks yang berbeza. Right? Next question, in order dalam citric acid cycle. Let's see citric acid cycle. Okay, apa soalan dia tadi? Sorry, tak nampak soalan. Fumarate 2H atom. Sorry. Fumarate from succinate will transfer to FAD to form FADH2. Basically, both FAD and succinate undergo redox reaction. Succinate and fumarate. Transfer to 2H will transfer to FAD to form FADH2. Okay. Betul. Succinate will donate the electron as well as hydrogen. So dia akan undergo oxidation. This one undergo oxidation. This one undergo reduction. Betul lah, dia adalah redox reaction. Even though it is a redox reaction, you need to identify which one undergo oxidation and which one undergo reduction. Alright. Okay, so let's move on to stop sharing dulu. Okay, so let's nak move on, tak boleh move on sekejap let's move on to the next chapter. Okay, so saya share dulu pasir-pasir tu kejap-kejap lagi Alright, so let's move on to photosynthesis pula. Alright, I think in photosynthesis you are introduced to banyak graph. Okay, so this is one of it. Action, you ada action spectrum and absorption spectrum. So make sure you understand what does it mean by action spectrum, what does it mean by absorption spectrum. So yang ni menunjukkan kedua-dua graf. Action spectrum, kita ada absorption spectrum juga. Right? So kita ada 3 major pigment yang kita tunjukkan di sini. Right? Think you kena faham lah graf ni. And then you also need to understand that dalam chloroplast kita ada thylakoid. So at the thylakoid membrane we have photosystem. Okay, photosystem. So kalau kita tengok kalau kita tengok cellular respiration tadi, kita ada yang atas tu kita kata intermembrane space, yang bawah tu kita kata mitochondrial matrix. In photosynthesis, you need to identify which part is stroma and which part is thylakoid space. Okay, ni lagi tunjuk. Right, so you need to know the the structure of photosystem. Okay. Tak nak explain I think you need to know the structure of photosystem. What does it mean by photon heat pigment, macam mana heat, macam mana dia heat, macam mana apa yang transfer ni. What does it mean by this arrow? Arrow ni menunjukkan apa? Okay, arrow ni menunjukkan energy is transfer from one pigment to another pigment. Okay, bukan electron yang transfer but the energy is transferred. Right? Electron tak transfer lah. Alright, so next you also need to know that luar ni adalah stroma, inside ni adalah thylakoid space where we have high H+ concentration. In stroma we have low H+ concentration, and that's why I said nearly similar to oxidative level of phosphorylation, that electron is moving from one complex to another complex in the electron transport chain. The energy is released, however the H+ is pumping at different location. Yalah, satu kat mitochondria, satu kat cellular, satu kat chloroplast kan, so lain lah location dia. So in this case H+ is pump. Remember I told you it is pump atau transport. You cannot say diffuse. Right? Diffuse hanyalah bila dia passive transport. Yang melibatkan ATP synthase ni, yang ni adalah diffuse. Right? So H+ is pumped from stroma into thylakoid space. From stroma into thylakoid space. And then it will diffuse through ATP synthase from thylakoid space into stroma. Right? So so we have a linear electron flow. In dalam photosynthesis we have linear electron flow, and we also have cyclic electron flow, right? And linear electron flow, kalau you tengok dalam nota you sendiri, dia akan lalu ETC ni, and ada anak panah yang menunjukkan ATP. Tapi by right dia bukan directly, ATP is not directly formed by the linear electron flow, tapi untuk formation of ATP mesti melalui proses redox reaction dan juga chemiosmosis tu juga. Lebih kurang sama macam lebih kurang sama macam oxidative phosphorylation tadi. Right? Tapi yang ni kita panggil sebagai photophosphorylation. Right? Okay. Saya rasa saya tak explain lah satu-satu Wah. Okay lah, saya explain satu-satu lah kat sini sekejap Kita explain satu-satu. Right, so saya rasa yang ni menunjukkan linear electron flow that kita ada dua photosystem, PS2 and PS1, which reaction center untuk pair of chlorophyll A. The pair of chlorophyll A in PS1 is P700. Pair of chlorophyll A in PS2 adalah P680. So P680 merujuk kepada the pair of chlorophyll A yang duduk dekat tengah-tengah ni. This is the pair of chlorophyll A. Duduk kat tengah-tengah ni. So this pair can either be It can either be P680 atau P700. Okay, so untuk P680, the photosystem is called PS2. For P700, the photosystem is called PS1. Right, don't get confused. PS ni merujuk kepada photosystem. P680 atau P700 is referred to the pair of chlorophyll A. Right, so I think to simplify the linear electron transport adalah the first one is light heat pigment. The energy is transferred from one pigment to another pigment until it arrives at P680, and then as P680 receive energy, right, the electron will excited and it will be received by primary electron acceptor. So P680 then become P680 plus. Right, so as it becomes P680 plus, it is a strong oxidizing agent. Right, so H2O will split. It will form H2O. As H2O split, it will form H plus, 2H plus, 1 oxygen atom and 2 electron. And these 2 electron will be donated to the P680 plus 1 by 1. Right, so this is what I'm telling you, that the difference between oxidative level phosphorylation dan photophosphorylation is the source of electron juga. Okay, sebab untuk ni source of electron dia adalah daripada H2O. Tapi H2O ni hanya akan split bila P680 ni jadi P680 plus. Okay, dan P680 plus ni hanya boleh P680 hanya boleh excite electron bila dia receive energy. Dari mana datang energy? Daripada photon of light. Okay, so I think the electron fall down, I think similar lah, electron fall down, electron transport chain, energy is released. Energy release is used to transport H plus from stroma into thylakoid space. Okay, and then the H plus will diffuse through ATP synthase, and then ATP synthase will catalyze the formation of ATP from ADP plus PI. Right? Macam itulah. Right? So linear electron flow will produce ATP via chemiosmosis dan juga akan produce NADPH. Okay? So these two things, ATP and NADPH, will be used by Calvin cycle, right? And we also have cyclic electron flow that the only ATP is formed sebab electron daripada P700 excited and then akan receive dekat ferredoxin. Okay, so ferredoxin is a mobile carrier. So kalau you tanya saya, kalau tidak ferredoxin kat sini, macam mana dia move kat sini? Jawapan dia, ferredoxin memang boleh bergerak sebab dia mobile carrier. Kalau you tengok dalam video, mana-mana video, you can see that ferredoxin can move. I think sama macam cellular respiration, kita pun ada mobile carrier dalam mitochondria juga. Okay, so dia akan bergerak. Right, so next one is Calvin cycle. So even though Calvin cycle do not need any light, however indirectly Calvin cycle need product of the light reaction. So kalau tak ada light reaction, Calvin cycle also cannot function sebab tak ada product daripada light reaction. So you can see in Calvin cycle, kita akan gunakan ATP dan juga NADPH, right? So Calvin cycle is divided into 3 phase. Phase 1 carbon fixation, phase 2 reduction, and phase 3 is regeneration of RUBP. Right? I think I tak explain semua, so saya cuma nak pecahkan dia saja kepada 3. Okay. Right, so carbon fixation is when we fix CO2 into RUBP. So you akan form intermediate first and then the intermediate split. Right, and then untuk reduction, of course nama dia reduction phase, memang akan ada proses reduction and oxidation, which your NADPH akan oxidize, donating the electron. Okay, so dia akan form NADP plus. And then dia donate electron kepada 1,3-biphosphoglysierate. So this one akan reduce. Right? And finally adalah regeneration of RUBP. So kita nak regenerate the carbon acceptor. Right? So next adalah untuk photorespiration. That photorespiration is basically a wasteful process. So in order to overcome photorespiration, some of the plant adapt to C4 pathway dan kita ada CAM pathway. Right? So untuk C4 pathway, the leaf anatomy is different from C3 plant. Okay, leaf anatomy is different from C3 plant. That we have, C4 plant have mesophyll cell and also bundle sheath cell. Mesophyll cell and also bundle sheath cells that conduct photosynthesis. Right? So kalau C3 kita hanya ada mesophyll cell, kita ada mesophyll palisade and spongy mesophyll, but hanya mesophyll saja yang mempunyai chlorophyll. Bundle sheath cell tak ada chlorophyll. Tapi untuk C4 leaf anatomy, bundle sheath cell will also conduct photosynthesis, which dia conduct Calvin cycle. Right? So in mesophyll cell, dalam mesophyll cell kita akan conduct C4 pathway, which CO2 is incorporated into PEP. Right? And it will form oxaloacetate. Right? And then it will form malate. Malate will move into bundle sheath cell and dia akan separate into pyruvate and CO2. Right? And we have additional enzyme which is called as PEP carboxylase. Right? So do we have Rubisco? Jawapan dia yes lah, sebab kita ada Calvin cycle, so we still have Rubisco. But in this case, Rubisco is located in bundle sheath cell. Right? So Rubisco will always receive high amount of CO2 daripada malate. Right? Sebab CO2 has been fixed in mesophyll cell first. Right, and this is the CAM pathway. Saya rasa yang dalam lecture note agak berbeza tapi lebih kurang sama je, that during the night the stomata open, so kita akan fix CO2 pada waktu malam sahajalah. Waktu siang tak boleh fix CO2. Okay. Tapi light reaction tak boleh berlaku pada waktu light reaction. Itulah, light reaction tak boleh berlaku waktu malam. So ATP dan NADPH tu tak adalah tak boleh adalah waktu malam sebab light reaction tak ada waktu malam. So you cannot conduct Calvin cycle during the night sebab tak ada light reaction. Okay, so apa yang you boleh buat, apa yang this CAM plant boleh buat adalah fixing the CO2 during the night. Kenapa dia nak fix waktu malam? Because waktu malam je stomata dia open. Waktu siang dia tutup stomata. Kenapa dia tutup stomata waktu siang? Sebab ada photorespiration. So dia tutup lah stomata. So during the night it fix CO2 Sama je sebenarnya pathway macam yang ni, fix CO2 dalam PEP by PEP carboxylase, dia form oxaloacetate and then dia form malate, right? And you can see malate is stored in vacuole as organic acid, right? And esoknya barulah malate tu keluar balik daripada vacuole masuk dalam chloroplast and then dia akan separate into pyruvate and CO2. Right, so basically chem plant as well as C4 plant are nearly similar. Both ada PEP carboxylase, both are fixing CO2 into PEP first and then baru malate akan differentiate atau break down into pyruvate dan juga carbon dioxide. But the difference between chem plant dan juga C4 plant is chem plant fix CO2 at different time. However, C4 plant it fix CO2 at the same time but at different cell. Okay. So far ada soalan? Why non-cyclic electron flow is significant? Non-cyclic is significant to provide, soalan ni non-cyclic is significant to provide extra ATP needed for Calvin cycle. Okay. Is it this live will see? I think it will be safe kot dalam YouTube. I think so. Lah. Let's say kalau dia tak safe, let me know. Okay. So let's move on to chapter 7, biodiversity. Tak banyak pun. Satu slide ya. Right, so I think in biodiversity you learn about taxonomy. Dua benda major yang you learn. One is taxonomy, the other one is biodiversity in Malaysia. So taxonomy ni adalah favourite question. Sama je sebenarnya, dua-dua pun favourite question juga. Selalunya dua-dua benda ni akan ditanya jugalah dalam final. Alright, so dalam taxonomy you learn about binomial nomenclature. So in binomial nomenclature, apa yang you kena tahu adalah cara menulis binomial nomenclature. Dia ada dua part. You nak italic ke you tak nak italic. So in this case kalau you type dia, pastikan you italic lah. Kalau you tidak type, you tulis tangan, pastikan you tulis, tak kisah you tulis tegak atau italic, tapi you mesti gariskan. Alright, nama spesies. Kalau nama genus, you akan italic juga, tapi kalau nama lain, dia punya takson yang lain, you tak gariskan lah. Right, next one is the importance of taxonomy. Yang ni I think you boleh tengok dalam lecture note saja. Next, you learn about hierarchical classification. This is hierarchical classification. You belajar hierarchical classification ni, you belajar dia punya taxon. Starting daripada kingdom sampailah kepada species. Right, and you learn about this 3 domain 6 kingdom system. So 3 domain 6 kingdom system. So these are the 3 domain: bacteria, archaea and eukarya. And these are the 6 kingdom system that we are using now. Eubacteria, archaea bacteria, protista, fungi, plantae and animalia. Right? And next one you learn about phylogenetic tree. At least apa yang you perlu tahu dalam phylogenetic tree ialah bagaimana apakah maksud branch tu, dan at least apa yang you kena tahu adalah siapa yang paling dekat dengan siapa, which organism that is closely related to the other, yang mana satu yang lebih dekat macam tu. Next one is biodiversity in Malaysia. You learn about the level of biodiversity, species, genetic and ecosystem diversity. You learn about the importance, the threat of biodiversity and the conservation of biodiversity. So I think yang ni tak susah sangat sebab you buat presentation tu basically you knew. Okay. apa yang you kena tahu, biasanya soalan mungkin akan bagi contoh dan you mungkin perlu memberikan jawapan based on contoh yang diberilah, which I think you are familiar with it. Okay, right, so I think let's move on to ecology. Dalam ekologi ni yang you belajar. Nampak ke? Saya zoom sekejap lah. Okay, so dalam ekologi you belajar tentang ecological hierarchy. Untuk ecological hierarchy ni saya rasa you belajar tentang species, population, ecosystem, macam tu lah, saya pun lupa dah. Okay, so make sure you tahu bagaimana nak define setiap hierarchy tu. Okay, what does define species contohnya, soalan yang biasa akan diberi. Define ecosystem, define population, macam tu. Right. Next one, you learn about ecological concept. In ecological concept, you learn about ecological niche and interaction. So basically ada banyak interaction yang you belajar, okay, ada banyak interaction yang didedahkan, including mutualism, commensalism, parasitism, okay, ada banyak interaction yang didedahkan. Namun kebanyakan daripada perbincangan berkisarkan tentang competition dan predation. Okay, kalau kita dapat lihatlah. Right, so in competition, alright, you are introduced to interspecific competition and intraspecific competition, and you are also introduced to the outcome. Right, so basically when we have and then you are also introduced to, kat sini before interaction ni, you are introduced to ecological niche. Okay, which niche is the role of individual in that ecosystem, in that particular area, the role of that individual. Alright, so it comprises of dia punya active time, dia punya location and resource yang dia guna. So tiga benda tu adalah ecological, tiga benda tu membentuk ecological niche, and you should know that the more the niche overlap, the higher the competition will be, right? So competition boleh jadi inter atau intraspecific. So kalau intraspecific, usually competition dia adalah tinggi jugalah, sebab kalau sesama spesies yang sama, ecological niche tu akan overlapping kesemuanya. Mostly dia akan overlap, right? Jadi intraspecific ni competition dia agak tinggi. Okay, however interspecific competition juga boleh berlaku sekiranya ada niche overlapping. So that will result in the outcome. Okay, sebab interspecific competition ni berlaku, so kita akan ada outcome dia. Alright, so by principle, the two competing species cannot coexist. Tak boleh hidup sama-sama. Dua species yang berbeza, okay, tak boleh hidup sama-sama. Itu prinsipnya. Right, I think that will result in competitive exclusion. Okay, yang I think you diberi contoh dua spesies paramecium, Caudatum dengan Aurelia kot, lupa dah nama dia. So which satu spesies tu, dua-dua spesies kalau letak dekat tempat yang berbeza, dia akan, graf dia akan meningkat lah, maksudnya population dia akan bertambah. However, kalau letak kat tempat yang sama, satu spesies akan menurun, satu spesies lagi population dia akan meningkat. Tapi peningkatan dia tak sama macam kalau dia duduk dalam keadaan perasingan. So that is competitive exclusion, which memenuhi prinsip that dua spesies tak boleh coexist at the same place. Right, sebab satu spesies akan basically being excluded, and finally mungkin akan extinct daripada kawasan tersebut. Alright. However, if that two species wanted to coexist, dia perlu ada adaptation. Okay, so it must do resource partitioning that will later eventually leads to character displacement. Kalau dia nak coexist. So this resource partitioning will result in fundamental niche and realized niche. Right, so fundamental niche adalah niche yang dia sepatutnya boleh ada. Okay, asalnya dia boleh ada, tapi realized niche ni adalah niche yang dia ada pada ketika itu during competition. Waktu competition berlaku, that is the realized niche, right? So I think for resource partitioning, contoh yang diberi dalam kuliah adalah the 7 lizard species that live on the same location, tapi dia duduk dekat different niche area. Okay, ada satu yang duduk dekat shaded area, satu lagi duduk dekat exposed area, ada yang duduk dekat fence macam tu. Right, so that is resource partitioning. Right, and for character displacement, you will see the example of the Galapagos finches that the character displacement occur due to evolution. Evolution, right? So eventually character displacement ni sebenarnya adalah hasil daripada resource partitioning, that the two species of bird, they eat different seed, seed. Eventually mula-mula dia sebenarnya makan seed of similar size, tapi sebab dua-dua duduk kat tempat yang sama, competition occur, so dia makan seed at different size. So eventually pada due to evolution, organism yang makan seed yang bersaiz besar mempunyai beak yang bersaiz lebih besar, sebab kalau beak size dia kecil, dia tak boleh makan seed yang besar. So eventually bila dia tak boleh makan seed yang besar, dia akan matilah, sebab dia akan compete dengan the other species yang makan seed yang kecil. Which yang ada big size yang besar, dia akan makan seed yang bersaiz besar, competition dia kurang, so dia boleh hidup dan dia boleh ada offspring, dan dia boleh menurunkan dia punya karakteristik. So eventually due to ovulation, after a very long time, you akan dapati ada perbezaan ciri antara spesies tersebut apabila berlaku competition. Okay, so usually character displacement kita kata is the results of resource partitioning due to evolution, after a very long time, right? Next interaction that we look quite deep adalah predation, that we look into the prey behavioral defense. Okay, so kita ada macam-macam behavioral defense, ada aposematic coloration, cryptic coloration and so forth, right? Next one, you learn about population ecology. So in population ecology, we look at characteristics, demographic and population growth. So untuk karakteristik, kita lihat population size, population density and population dispersal. So population size is the number of individual. Tak boleh zoom, tak boleh conteng. Alright, so population size is the number of individu, and ada 4 faktor yang boleh mempengaruhi population size. They are birth, death, immigration and emigration. So basically these 4 faktor juga boleh mempengaruhi population growth. Alright. Next one is population density, that it is also the number of individual tapi per unit area. Okay, kepadatan dia. Next one is dispersal. So dispersal ada 3, clump, they change over time. So it is the study of vital statistic. So basically the demographer are interested on the life table, the survivorship curve and the reproductive rate. Survivorship curve ada 3 type, type 1, type 2 and type 3. I think type 1 adalah human. Mati, kebanyakannya mati pada umur yang tua. Type 2, death rate dia sama throughout the lifetime. Untuk yang ketiga ni adalah plant, that the death rate dia adalah tinggi waktu seed, waktu umur yang muda, tapi death rate dia menurun pada umur yang lebih dewasa. Right? And next you learn about population growth. So population growth is the number of changes towards the number of individual over time. Masih lagi melibatkan number of individual, tapi beza dengan size ni, size tu adalah pada masa tersebut. Population growth ni kita melihat over time. Okay, over a period of time. Sama ada number tu menurun atau meningkat. Right? So sama, dia masih melibatkan 4 faktor tadi, birth, death, emigration and immigration. Right? And population growth is affected by limiting factor. So kita ada 2 types of limiting factor. Density dependent dengan density independent. Okay, berkaitan dengan dia terkait dengan density atau tak. Kalau yang terkait, dia usually akan melibatkan benda-benda yang melibatkan competition. Contohnya macam rumah, makanan dan sebagainya. Kalau yang tidak berkaitan dengan density, dia biasanya adalah natural disaster. I think contoh yang paling dekat sekarang adalah COVID-19. Okay, other than that, banjir semua tu juga adalah limiting factor. Alright, and next you learn about growth model. Ada 2 growth model, exponential and logistic. Right? So untuk exponential, yang ni kalau kita assume limiting factor sebagai zero limiting factor. Okay, dia boleh naik secara eksponen. Okay, logistik ni apabila ada competition lah, apabila ada limiting factor. Right, finally you belajar pasal biogeochemical cycle. So carbon cycle adalah gas cycle, phosphorus adalah example untuk sediment punya cycle. Right, so I think ini saja yang you dah belajar, carbon cycle, phosphorus cycle. So I think inilah yang you telah belajar throughout the semester. Resource partitioning. Okay, sekejap soalan ni, kita baca soalan sekejap. Possible tak tanya biogeochemical cycle other than carbon cycle? Tak possible. Soal adakah possible untuk tanya cycle yang lain? Tak possible lah. Biasanya itu saja cycle dia ajar, tu je lah yang ditanya. Explain about resource partitioning. Okay, boleh? Kejap je lah ya, kita explain pasal resource partitioning. Sekejap ya, saya cari. Okay, tutup yang ni sekejap. Okay, alright, saya share screen. Kita explain pasal sedikit tentang resource partitioning. Okay. So let's look at, salah. Okay, sebelum saya explain pasal resource partitioning je. Think I'll explain about ecological niche first. Okay. Okay. Ecological niche. the ecological niche first. Sebab in order to understand the whole concept of competition, kena tahu ecological niche first, okay? Kalau saya salah, tolong betulkan saya lah, kot-kotlah saya salah kan. Ni sefaham saya lah ya. Ecological niche is refer to functional role, okay? Peranan you, okay? And dia comprise of these three: habitat, kat mana dia duduk, period of time, bila waktu dia aktif, dan the resource yang dia guna. Resource yang dia guna. So that is ecological niche, right? Contoh, as a student, you live in pusat asasi, right? So that is your habitat. Habitat you di pusat asasi, right? Period of time, bila you aktif, okay, contoh pukul 10 pagi baru you bangun untuk cari breakfast. So that is time yang you aktif. Okay, daripada pukul 12 tengah malam sampai pukul 10 pagi you tidur. Okay, ataupun dari 10 pagi semalam je sampai 10 pagi ni you tidur 12 jam. Okay, you tidur. Macam baby kan, 12 jam you tidur. Okay, dah pukul 10 baru you bangun cari breakfast. So that is your period of time when you are active, right? And you utilization of environmental resource ni contohnya you makan nasi goreng. So basically semua orang kat pusat asasi tu makan nasi goreng sebab nasi goreng yang murah dan sedap, contoh. Right? So you have ecological niche that you live in the same area, pusat asasi. Period of time when you are active adalah sama. Waktu dia you aktif pada pukul 10 pagi, contoh dia. Okay, and you makan nasi goreng. Tak nak makan makanan lain, bihun goreng semua tak nak, semua makan nasi goreng. So basically we have student dekat pusat asasi tu contohnya mempunyai ecological niche yang sama, habitat yang sama, period of time, active time sama, makan pun benda yang sama. So the niche yang overlap adalah tinggi, maksudnya semua sama, niche yang overlap tu semua sama. So competition dia adalah sangat tinggi, which pada pukul 10 bila you keluar bilik, semua orang keluar bilik waktu yang sama, semua orang cari nasi goreng. Right, competition dia jadi sangat tinggi. Right, so what do you need to do untuk you reduce competition tu? Jadi kita akan buat resource partitioning. Okay, kita akan buat resource partitioning. Apa yang kita boleh partitionkan? Okay, kita boleh partitionkan waktu kita aktif. Boleh lah, okay, as a human kita boleh buat partition waktu aktif. Contoh, aku bangun pukul 10, kau bangun pukul 11, kau bangun pukul 9. So kalau kita dah partitionkan waktu aktif tu, so kita dah mengurangkan competition. Apa lagi yang kita boleh buat partition? Kita boleh partitionkan kita punya resource. Tadi semua orang makan nasi goreng kan? Okay, instead of, oleh kerana semua orang kena makan nasi goreng juga, okay, why not kita partitionkan? Okay, satu kumpulan akan makan nasi goreng daripada gerai nombor 1. Satu kumpulan makan nasi goreng gerai nombor 2. Satu kumpulan makan nasi goreng gerai nombor 3, contoh. Jadi kita dah partitionkan masa, kita juga dah partitionkan resource yang kita ada. Betul? Jadi apa yang akan berlaku? Competition tu akan meningkat ke berkurang? Macam lah ada jawapan kan? Okay, competition tu akan berkurang sebab kita dah buat partition untuk masa dan juga resource yang kita ada. So similarly kepada contoh yang diberi kepada you dalam lecture lizard ni, right? Dia makan benda yang sama, which dia makan dia makan insects and small arthropod. Okay, dia makan benda yang sama. Jadi apa yang dia boleh lakukan adalah dengan divide dia punya area, niche yang dia duduk, area. So satu spesies duduk kat tempat lain, satu spesies duduk kat tempat lain. Masih lagi dalam habitat yang sama tapi area yang berbeza. Jadi dia dah partitionkan dia punya habitat. Okay, niche kat mana dia duduk. Mungkin dia masih aktif pada masa yang sama, dia masih makan benda yang sama tapi dia dah partitionkan area dia. Boleh ke? So far, that is resource partitioning. Alright. so I stop sharing. Okay, kita dah live dah dekat 2 jam dah ni. Saya rasa macam saya takut tak sempat, tapi saya cuba kongsikan sedikit daripada pasir. Boleh tak tahu pasir pasir kejap pasir korang dah you all buat ke pasir yang dalam paddock tu? Saya rasa macam tak sempat untuk tunjuk semua soalan. Banyaknya respon. Macam tak sempat dah tunjuk semua soalan. Soalan mana yang susah dan perlu saya yang perlu saya explainkan jawapan. Saya rasa soalan ada banyak dan macam tak sempat lah nak explain semua jawapan. Boleh tulis dalam chat tak soalan yang mana satu? Cik. Kalau tak ada, mana okay, explain mana yang sempat je lah Right, so untuk soalan yang pertama, soalan tentang role of carbohydrate in plasma membrane. Alright, saya rasa soalan ni senang je, role of carbohydrate in plasma membrane. Betul, so as long as you letakkan role of glycolipid atau glycoprotein, they are correct lah. Okay, next one adalah okay. This one, how glucose cross plasma membrane by passive transport. So yang ni adalah using soalan tentang how glucose cross plasma membrane by passive transport. So I think untuk soalan ini jawapan yang expected from you adalah using carrier protein. Glucose is transported using carrier protein. The name of carrier protein is glucose transporter. Glucose transporter, and it is transported by facilitated diffusion, and of course you boleh kata down concentration gradient, do not use energy. Okay, apa-apa yang you tulis tentang passive transport tu adalah betul lah, right? Boleh ke saya baca je lah ya jawapan? Slide 33. Okay, jauh lagi tu slide tu. Right, next untuk vitamin K. Vitamin K is non-polar, right? So dia non-polar. So since vitamin K is non-polar, so it can dissolve through phospholipid bilayer by diffusion. And also you boleh tulis lah follow concentration gradient, do not use energy. I think let's go to slide 33. Sorry. Okay, Nick Mimi minta slide 33. So I think I skip to slide 33 dulu. Kalau ada request, tolong tulis dekat komen tu lah ya. Okay. Right, so I think this is the question 2019. Soalan tanya tentang apa nama dia? Phylogenetic tree. So based on the branch point, explain why clouded leopard and snow leopard belongs to the different genus. Okay, saya write down dalam chat slide 33. Okay, so you can say that it have different morphology atau characteristics atau sequence of DNA. The other point is right, so dalam saya tuliskan jawapan kat sini untuk slide 33. So soalan tanya pasal based on the branch point kan. So clouded leopard is this one and snow leopard is this one. So branch point dia berbeza. So I think the easiest answer that you can give adalah it have different common ancestor. Right, so the second point yang saya bagi, different common ancestor. Right. Next one, minta soalan nombor 7. Okay. Let's look at soalan nombor 7. Kita balik ke depan. Oh, okay. Seorang je student yang respon. Okay. Apa? Results of the investigation that support the idea that glucose enter the cell by facilitated diffusion. Okay. So kalau dari sini, soalan ini menunjukkan, okay. Let's look at the diagram. Boleh Mini, kejap ya, kita explain slide 7 dulu. So soalan ini, dalam rajah ni menunjukkan glucose uptake by the cell. So concentration of glucose, as you can see, bila concentration of glucose meningkat, glucose uptake sepatutnya meningkat. Tapi kita dapati bila dia meningkat, tiba-tiba glucose uptake ni menjadi menjadi setara, tiada peningkatan dalam uptake of glucose. Okay, so basically kalau dia bukan facilitated diffusion, graph dia sepatutnya straight lah. Kalau dia bukan facilitated diffusion, graph tu sepatutnya straight sebab facilitated diffusion akan menggunakan protein. Right, macam pintu lah, macam you ada pintu, kalau tak ada pintu semua orang boleh masuk. So tak ada sekatan. Contoh macam highway, sekarang PKP kan. So kalau tak ada sekatan, semua orang boleh jalan dekat jalan raya tu. Tak ada orang marah pula kan. Tapi oleh kerana sekarang PKP, so ada sekatan pada jalan raya. So what happen is orang tak boleh lalu. Maksudnya orang boleh lalu tapi dia lalu tu dalam keadaan yang perlahan kerana ada sekatan. Okay, so similarly untuk soalan ni, kalau tak pakai protein, sepatutnya sel boleh terus menyerap glukos tanpa menggunakan protein. Lalu je lah. Tapi glukos hanya boleh transported menggunakan facilitated diffusion. Dia mesti pakai protein. That's why dia at one point protein tu dah tepu dah. Maksudnya protein yang ada hanya boleh transport that maximum amount of glucose saja. Dia tak boleh transport lebih dari itu. Itu je kelajuan yang paling laju yang dia boleh transport. Bukan kelajuan. Itu je jumlah yang dia boleh transport. Maksudnya contoh kalau satu sel tu dia ada 10 carrier protein untuk glucose. So dia hanya boleh transport 10 glucose pada satu-satu masa. So rate of uptake tak boleh tinggi. Okay, so bila you tengok rate of uptake dia sama, dia bukan bermaksud glukos tu tidak diambil ke dalam sel. Glukos masih diambil ke dalam sel tapi pada kadar yang berbeza. Pada kadar yang sama. Okay, so glukos masih diambil masuk ke dalam sel tapi kelajuan dia itu je. Dah maksimum dah kelajuan dia, tak boleh laju lagi, tak boleh banyak lagi ambil. Right, so jawapan yang you boleh tuliskan ialah ialah dengan you describe graph ini. Cik. Ada orang jawab dah kat sini. Mari kita tengok jawapan. Tak jawapan. Boleh lah maximum uptake of glucose. Okay, so kita boleh jawab. Okay, ni slide berapa? Slide 7 kan? Slide 7 Slide 7. So kita boleh kata pertama sekali, as concentration of glucose increase, rate of glucose uptake is also increase. Kejap. Itulah. Is also increase. However, then it becomes plateau. Then it becomes constant. Maximum amount of glucose Alright, so I think ni lah jawapan dia. Concentration of glucose increase, rate of glucose uptake also increase. Tapi it become constant. So it shows that maximum amount of glucose has been transported by transport protein. Okay, sebab soalan tanya macam mana kita nak tunjukkan yang dia adalah facilitated diffusion. Right? Okay, so let's go to slide number 19. Oh, okay. Right, so let's go to slide number 19 from 2018. Okay, so soalan tanya, what effect does this action have on cellular respiration? Okay, so kalau dia akan form complex dengan cytochrome, and then it prevent electron to transfer into final electron acceptor. Right, so slide 25, boleh. So what happened is bila cyanide prevent the transfer of electron kepada electron acceptor dalam ETC, first sekali ETC akan dysfunction lah. ETC akan dysfunction which no electron transfer occur. So kalau electron transfer tak occur, you cannot transport H+, you cannot transport H+ from one matrix to intermembrane space. So you cannot produce proton motive force. So kita kata no proton motive force is generated. Right? And then bila tak ada proton motive force, so ATP tak boleh produce lah. So kita kata no ATP produce. Right? And then Kalau tak ada ATP, kalau sebab dia kata dia tak bagi elektron pergi dekat final electron acceptor kan. Final electron acceptor is oxygen. So oxygen akan combine dengan H plus, so dia akan form H2O. So eventually no water is produced. No water molecule will be produced. Right, so I think this is the answer for slide 19. Okay, so let's move on to slide number 19. Slide apa lagi? Slide 25. Oh betul, susah soalan ni kan? Sebab ada seorang je yang ni. Soalan tanya the advantage of the new rice. Betul? This answer is correct. Congratulations. Siapa yang jawab ni? Oh tak ada nama Congratulations to anyone that answered this question. Your answer is correct. The new rice can live in extreme hot and dry environment, atau you kata it can it able to stand hot and dry day, right? And then betul, it can carry out C4 pathway that it incorporated CO2 into PEP. It can minimize photorespiration. Okay, so jawapan ni betul jawapan yang lain yang boleh ditulis di sini slide 25. Okay, so yang pertama, able to stand hot and dry day. Yang kedua, you boleh kata minimize photorespiration. Yang ketiga, estimated yield increase. Yeah. Okay, the rest dah ditulis lah, boleh fix CO2. Right, ni lah jawapan lain yang you boleh berikan. Able to stand hot and dry day. Minimize, saya tak letak titik lah kat situ, sorry lah dia jadi sambung terus, tak malah ya. Minimize photorespiration, estimated yield increase. Right, so jawapan estimated yield increase tu kenapa boleh you tulis, kerana this is rice. Okay, so if you can minimize photorespiration, you akan produce lebih banyak G3P kan? Okay, so you akan produce lebih banyak sugar. So lebih banyak sugar maksudnya lebih banyak carbohydrate yang you boleh produce lah. So the estimated yielding akan increase. Other than that, it has been write down by your friend. It can it can fix CO2 efficiently lah. Sebab dia pakai PEP. Alright. Okay, so far I think tak ada soalan lain dah. Okay, ada soalan lagi? Kalau tak ada, I think kita stop di sini. It's nearly 4 o'clock. Selesai dah kot. It's nearly 4 o'clock. So, saya rasa selesai dah 2 jam untuk untuk kita punya sesi live revision hari ini. Thank you very much for attending this virtual class. Okay, so hopefully that it benefit you well. Okay, and don't forget to fill in your attendance. Okay, I think ada dalam komen atas sekali kot, jadi dekat atas sekali. So don't forget to fill in your attendance. Right, so kalau tak ada apa-apa, with that saya rasa kita tutup dengan tasbih kifarah dan suratul Asr. Right, so boleh tutup lah dengan doa tamat pembelajaran. Dan saya doakan semoga semua berjaya jawab dengan baik, dan kalau ada soalan lagi boleh tanya kepada pensyarah masing-masing, dan kalau student saya anda masih lagi boleh tanyakan soalan pada saya. Alright, and I think you boleh je menjawab dalam padat tersebut. I think kalau ada masa mungkin mana-mana pensyarah pun boleh kot rasanya akses kepada padat tu, kita boleh betulkan je. Alright, so thank you very much. Assalamualaikum. Bye.